In today's episode, we'll be going through nucleotides and nucleic acids topic. So the questions I'm going to go through, which are these, they are essentially the same really, but just written in a different way. With these specific ones, however, I require you to actually know the practical before we answer this. Let's begin. So, remember, first exam technique is read the question first. So it says, suggest why the cells do not need to be crushed before adding detergent. Okay, let's actually read the information now. So DNA can be extracted from a culture of white blood cells and precipitated using the following procedure. Mix a culture of white blood cells with a detergent. So why does it not need to be crushed? Well, this is going to be some application, sort of question, only because just by looking at the information given, I can't really tell you straight away why it doesn't need to be crushed. I think it may have to do with the fact that it's about white blood cells. Okay, They're cells and they aren't tissues. It's in the name, right? So I guess it doesn't need to be crushed for that, because the cells are there. You don't need to get them, out. Okay, next question says, explain why the detergent is used in step 1. If you've done the practical, you would know that that's all about dissolving the phospholipid bilayer. Next one says, suggest the type of enzyme that should be used in step 3 and explain why. So 1 mark for the name of the enzyme and the 2nd mark for the explain why. So this is about enzyme, and we are talking about DNA to be extracted. So DNA 101. DNA is in a double helix structure, and that's awful drawing, but is bounded by histone proteins. And well, as that is a protein, and you need to essentially extract the DNA, I'm going to be adding protease because it will break down the histone proteins because it's in the name. I don't understand terms. For example, white blood cell. It's called a cell. It's not an organ, it's not a tissue, it's a cell, right? So that's where this point comes in. Not many people would actually think about that. I'm assuming people would think of this point, but rarely would anyone think that one, only because it might be considered a bit of a stretch. But the marks team loves it, so that's fine. And yeah, the rest we got correct. Okay, that's great. Next one. Let's read the question. State the purpose of step 1. Right, well, what is this question about? Purifying some DNA from leaf cells. So you want to grind it. Well, what's a leaf? It's a plant. What do plants have? They have cell walls. The purpose of grinding it is to break down the cell wall. Suggest why a protease enzyme added in step 3 is needed to purify DNA. Well, we spoke about it in the first question, so we can all guess what the answer is. Yep. We got them correct. Great. Next one. Suggest why this would not be an appropriate source of protease when attempting to produce a pure sample of leak DNA. So it's saying that the students considered using pineapple juice as a source of protease enzyme. Oh my god, that is awful. No, that is wrong. If you want to get, and I quote, a pure sample, then surely you can't be mixing 2 sets of DNA, right? We can all agree that everything has DNA. Pineapple juice has DNA. So if you're using the protease enzyme in the pineapple juice, then doesn't that mean you're mixing both those DNAs together? So then surely that's not a pure sample. Stress one important step that the students had left out of their method. immediately I'm thinking detergent. Because I've done so many of these like types of questions where it has this practical in particular, I know that you need detergent, and it hasn't mentioned that. Like, look here, it says detergent, right? Does it say detergent here? No. So that's straight away my answer. Only by understanding and knowing how the practical is. Oh, one very important thing is that if you're ever stuck in the exam and you're like, oh god, like, I don't know what the answer's gonna be, and it's a practical question, well, just don't try and think of like a textbook or mark scheme answer. Worth doing is just retrace your steps of when you were doing the practical in class. Like me, I remember, oh yeah, I added detergent. Did they say it here? No, they didn't, right? That's my answer, great. One mark, you know? So relying on your intuition and your gut feeling. Name the process described in step 6. A white layer actually when I read that, when I was reading the steps and I got to this part, I was about to say the answer. If you know the answer before I say it, then that's great, but let's read it again. A white layer of DNA forms between the sample and the ethanol. That is called precipitation. You just need to learn it. Okay, let's check the answers. Yep, we got them all correct. Oh, that's quite an interesting point they said here. Idea that pH might be too low. Yeah, okay, that would make sense. Pineapple is very acidic. Have you noticed how when you eat it, like your tongue has that like strange sensation? Your like lips feel wrinkled in a way. So yeah, that's quite a valid point. I don't know if I would have said that, but this is just a very, very safe answer. That is how you answer these questions. Now remember the two most important exam techniques are read the question first and state all the obvious points. So now it's your go. Try some questions and let me know how it goes.